My name is Munira and my partner's name is Sarah, and today we will be reviewing how liver enzymes are associated with hepatic insulin resistance, insulin secretion, and glycogen concentration in healthy men and women. The aim of our article was studied to determine whether liver markers GGT and ALT are mainly associated with peripheral insulin resistance, hepatic insulin resistance, and insulin secretion in a large cohort of healthy men and women participating in a relationship between insulin sensitivity and cardiovascular disease. The pathophysiological mechanism to illustrate the link between the risk of type 2 diabetes to highlight the concentration of GGT and ALT. Information such as smoking, physical activity, height, body weight was obtained, as well as alcohol consumption, which were calculated in grams per week. For the oral glucose tolerance test, also known as OGTT, blood samples were taken before and at 30 minutes, 60 minutes, 90 minutes, and 120 minutes into the OGTT, collectively with samples for central analysis of routine blood chemistry. Blood collected during the studies was separated into plasma and serum, liquid and stored, at -20 Celsius for glucose and at -80 for lipids.
In Table 1, men fasting glycemia were more strongly linked with GGT than ALT activity. In women, fasting glycemia were more strongly linked with ALT than GGT activity. In both sexes, GGT was linked with 2H glycemia even after putting in consideration factors such as age, center physical activity, alcohol intake, and waist circumference. In women it was 0.04 and in men 0.037. In contrast, ALT activity was no longer related with 2H glycemia in either men that had 0.08 or women that had 0.10. In multivariable models after further changes for the difficult factors mentioned above. In Table 1, fasting glycogen level was significantly linked with ALT concentrations in both men and women after adjustment of age, physical activity, alcohol intake, and waist circumference. The relationship between fasting glycogen and GGT level was weaker, and the association remained significant in the multivariable model in men only. In both men and women, the association between fasting glycogen and ALT persisted after adding BMA and waist circumference in the multivariable model, which was 0.01 in men and 0.003 in women. This association was independent of GGT activity, MI, fasting glycemia, and fasting insulinemia. In Table 1, basal and total insulin secretion rates were significantly associated with both liver markers. This association continued after adjustment for MI. In Table 2, insulin secretion increased across quartiles for GGT after adjustment of age, physical activity, alcohol intake, waist, and MI. If we look back at Table 3, a similar pattern was observed for ALT in men, but did not reach statistical significance in women. These relationships between liver markers and insulin secretion persisted after exclusion of participating with GGT or ALT values above the normal range in both men and women. In Table 1, both endogenous clearance of insulin and hepatic insulin extraction during the OGTT were negatively associated with GGT activity after adjustment of age and recruitment center. In Table 2, both decreased across quartiles of GGT in men and women in the multivariable model. In Table 3, there was no association between insulin clearance and ALT activity in the multivariable regression. In Table 1, the insulin sensitivity both men and women had a higher ALT or GGT activity, which was significantly linked with a lower MI after the change of age and center. This association was unchanged in the multivariable models. If you now refer to Table 2, insulin sensitivity decreased across the quartiles of both ALT and GGT after adjustment of age, physical activity, alcohol intake, and waist circumference. In conclusion, our article study shows that in healthy men and women increase the GGT and ALT activities within their physical range are associated with peripheral but also hepatic insulin resistance, increased insulin secretion, and decrease in hepatic insulin clearance. Furthermore, ALT activities were more positively associated with fasting glycogen concentrations, independently of the insulinemia, waist circumferences, or insulin sensitivity. These findings confirm the role of the liver in the pathogenesis of type 2 diabetes. And the novel findings of a positive correlation between ALT and fasting glycogen concentrations needed to be confirmed in type 2 diabetes, which means for now it is not proven. Thank you for listening.